I said that out here in the cell, in the cytoplasm, is where glycolysis happens. If you have oxygen, then the rest of it takes place in the mitochondria. If you don't have oxygen, then fermentation will happen also out here in the cell cytoplasm. Only thing happens inside there is respiration if you have oxygen. They'll go in there and then give you tons of ATP. Okay, now, this is Krebs Cycle. It's just one half of what's happening inside the mitochondria. Now, this will happen in the mouth. If you look carefully, there's not much going on. I mean, this is where you give off carbon dioxide and you breathe it out. Pyruvic acid comes in, and I'm just going to talk for a minute here, and it's converted to Acetyl CoA, which goes this way. Now, pyruvic acid, remember, has 3 carbons. We just lost one right there. How many carbons are in Acetyl CoA? 2. All right. Now, this thing right here is going. There's a compound coming this way, and this compound, they merge, they join to make this one. And this one is reshuffled to make that one. Okay? And then the numbers, those are steps. Second thing, third thing, fourth thing, fifth thing, sixth thing. That's all they mean. Okay? This will be reshuffled to form this. And then here now, you're going to give off some more carbon dioxide, you're going to breathe it out. But what you're going to do, you're going to give off an H product. That H represents energy that you can use. But you got to grab it. I tell you, that NAD, nicotinamide adenine dinucleotide, NAD, is a catcher. It will hold things for you. So when Krebs rips off the hydrogen, he throws it to the NAD and says, here, hold this. And NAD grabs it. Here is grabbed it. Now, you just lost another carbon. Now, this is 4 carbons joining 2 carbons. So what's 2 plus 4? 6. He has 6. That thing happened here. He has 6. Oh, carbon's gone. He has 5. What you did here, you're going to do again. You're going to release another carbon and get some more hydrogens. And now he had 5, so he has 4. Now, come through here. This is a form of ATP. So you're going to get, if phosphorus comes in, you're going to make this substance. This is still 4 carbons. No carbon dioxide come off, that's still 4 carbons. Now FAD is another catcher, but FAD can catch 2 hydrogens. So, and these H's are going to go to someplace else and do their thing, the electron transport thing. Now, there's still 4 carbons, and he's reshuffled, like shuffling a deck of cards, just reshuffle same cards to produce this. You're going to tear off another hydrogen. You got to have NAD to grab it, so it grabs it. And this is a 4 carbon, and you're right back where you started. This 4 carbon, and this is cycle, and it never ends. So I'm going to give off 1 carbon, 2 carbon, 3 carbons in this process. Now let's take it and give you what's going on. The matrix is not the membrane. Let me back up a minute. You see the blue right there? That's the matrix. That's the matrix. And what I just showed you, Krebs cycle, or it's also called the citric acid cycle, happens in the blue. Because this is called the matrix. That's where that's happening. It does not happen on the membranes. That's where the electron transport is happening. Another process. Now, pyruvate is a 3 carbon, right? And you split glucose to get 2 of those, right? Well, In the Krebs cycle, you're going to find that that glucose can only produce 2 ATPs, 1 per pyruvic acid. And you got 2 of them. And right there, I should change that to ATP. Because this is I can't recall what she stood for now. But I should go back and make that change. Because that is ATP. And if I have 1 given off per pyruvic acid, or 2 pyruvates per glucose, then Krebs cycle just gives me 2 in all. You with me so far? That's just 2 in all. So glycolysis gave me 2. This gives me 2. I'm going to get 38 in all, I thought. So the electron transport chain must give me the other 34. And it does. But all this does, I need those H's. I need those hydrogens for the transport chain. And so that's not bad. I'll get some ATP on the way. But the goal for Krebs cycle is not to make ATP, is to release 1, 2, 3, 4, 5, 6, 7, 8 hydrogens, 9, 10. That's going to then be used to make all the other energy. Okay? Now let's take it now, what I had here. Like I told you, this cycle happens, the blue circle in the matrix, not on those not the cristae. When I had you do your picture and label cristae, I wanted to let you know, those are the membranes. The cristae is the membrane where you find the ETC happening. This is not happening on the cristae. This happens inside the cristae in that inner compartment, all the blue area. Now, the pyruvates were produced with glycolysis because you split glucose in half, right? Now, with me so far, okay? Now, glycolysis, like I said, will only give you 2 ATPs net. Understand that? Mm-hmm. It took 2 to go in, 4 came out, so I'm hit by 2. Okay? But those pyruvates, if you have no oxygen, what process happens next? If you have no oxygen?
Fermentation.
Fermentation. And what are 2 types of fermentation? There's the lactic acid fermentation, and then there's the alcohol fermentation. Alcohol fermentation. Lactic acid happens in animal cells where you're running real hard. You don't have enough oxygen to carry out respiration, so you do that and you give out. The other kind of alcohol respiration, bakers use it for the carbon dioxide to make the bread rise. Brewers use it for the alcohol to make the drink. So there is no yeast fermentation. Yeast does do fermentation in both industries. And there's 2 byproducts. There's a carbon dioxide product that one industry loves, and the alcohol product that the other one loves. It's just the same thing. It's still alcohol fermentation. So the energy is used to make ATP. So the pyruvate molecule is going to be processed through the Krebs cycle. Pyruvate does not go through the electron transport chain. Those H's go to the chain that came from the Krebs cycle. Now, I showed that picture to you on the last page, so you'd have it one time. All right, step 1. Now I'm going to show you step 1, then show you a picture of it. Okay, step 1. Acetyl CoA came from pyruvate. Follow me out? Pyruvate, reshuffling, give off a carbon dioxide. So what you're going to find out is that pyruvate is a 3-carbon compound. It's changed to this by removing a carbon dioxide. Therefore, this is a 2-carbon compound. Okay? Now, when you remove the H ion, an electron goes with it. Then it's like a married couple. Where I go, the wife goes. So when I remove the H, I also get the electron, which is what I wanted. I can't move the electron, but it's like, like a dog with a piece of meat. Where the meat goes, that puppy's going to follow it. You can't pick the puppy up, so you just use the meat to move the puppy. Where the H goes, the electron's going to follow it. And the body knows this. All right? So, the when this is torn off, and hydrogen is also released, but it's got to be grabbed by this. And this plus is like your hand. And when this is made available, then this compound forms. And there's your hydrogen. It didn't get away. Oh, you breathe it out too? So it's going to be used later. And coenzyme A is what makes it all happen. And that's what we call this, when you join it with acetyl, you get Acetyl CoA, which is coenzyme A. Now, this is what goes into Krebs cycle. Let me back up and show you now. Right here, that's Acetyl CoA. And you notice you lost the carbon. You also lost the hydrogen with the electron that you wanted for the chain. And then this now enters this cycle. Okay? Now this is a blow up of just that one section. I took this section here and made it bigger for you. And you have it on your PowerPoint probably. You got to take a picture of it. All right. So this is coming from glycolysis. And because you have oxygen, then this travels into the mitochondrion. Now the mitochondrion now, this compound is changed to this compound. And in the process, 3 carbons are reduced to 2 carbons because you give off carbon dioxide. We're breathing out carbon dioxide. That's where it's coming from, some of it. But what you really wanted was these H's, because those H's come along with electrons. So when I tear off this compound, is nowhere near what this thing is. I mean, you've lost 2 H's, you lost a carbon. Now, here's Krebs cycle coming around. This is going to enter Krebs cycle. There's something coming this way. It's right there. There's something coming up. It's going to join together with this one. Now, here's my next step. And now I'm looking down right here. I'm right here. Now, this is called citric acid. Citrate is citric acid. And because that is the first compound formed in the cycle, we also call this the citric acid cycle. If the first thing had been some other acid, it would be called the other acid cycle. But since Mr. Krebs was the one who first laid this out for us, other scientists decided to name it in his honor, the Krebs cycle. So the Krebs cycle, citric acid cycle, same thing, same picture. So if you ever look up an image, and when I wanted this one, I first typed in Krebs cycle, didn't see anything I liked. Then I typed in citric acid cycle, and I found this picture. I said, oh, I like that one. Problem was, though, these things can't be read until I blow them up. And that light color there says 2C, so I wrote 2C on top of it. Because I want you to keep track of the carbons, how they're coming out. So this one is right there coming down. This one is that one. And I don't know these names, they're hard to know. Oxaloacetate comes around. It's a 4-carbon compound, this is a 2. So when 2 joins 4, you become 6. That's your citric acid. And for the longest time, this was called the citric acid cycle. Okay? Now, so this, you're back to a 6-carbon compound. Like glucose was, really. Okay? Now, something's going to happen. And my next arrow is going to be right there, but let me see. Okay? So the arrow right there in between these two. Now there's the citric acid. And this is isocitrate. That means different form, same atoms. Iso means same, right? So we just reshuffle the cards. If I give you 6 cards, and one time you have them arranged by numbers, next time you have them arranged by the suit. Same cards, right? Different appearances. Same atoms, different shuffles. Same 6 carbons. So right here, all we've done, we've reshuffled. That's going to expose hydrogen so I can get to it. So the citrate is converted to isocitrate, which is an isomer. It's the same chemical formula. And I don't know what that's there for. I won't bother now though. As another. Remember when we talked last about glucose being C6H12O6, but so was galactose, and so was fructose. Those are all isomers too. 3 different things having the very same C6H12O6 nomenclature. So if you knew what the formula was for this one, it'd be the same as that one. Just that they'd be shuffled different, same numbers. So all we have here is simply a reshuffling, and I'm looking right there, I'm looking right there. There's no H's coming off, there's nothing happening, it's just getting ready for this step. The next step, the air is going to come down here. And there it is, the air is down there. And this is step 4. This is step 4, I call it. Now, this iso is going to lose the carbon dioxide. Lose the carbon. Also going to lose a couple of H's with their electrons. Now, if this is 6 carbons, and I lose the carbon dioxide, that's got to be 5 carbons. And I don't know these names, I don't expect you to either. But I expect you to follow the carbons up. If I have no name here, and nothing here, and a question mark carbon, and a question mark carbon, and you see this, you should be able to track it on around and know how the carbons are changing, right? I mean, there it tells you that. If you think he's 5, and you say 4, you thought wrong, didn't you? I mean, you did it right, but you missed it. So if you start here correctly, and you know he is 3, you know this one is 2, you know 4 joins 2 to make 6. Nothing happens in 6, it stays 6. I'm right here. If a carbon comes off, then that 6 has got to become 5. Now, but now in doing that, we got that again. That's what you want right there. That's what you want right here, buddy. Because that's the whole purpose, is get those hydrogens and their accompanying electrons for the electron transport chain. All right, so now there's that, if I say ketoglutarate.
Alpha ketoglutarate?
Huh?
Alpha ketoglutarate.
Okay, now, we come down here. You chemistry folks all know this stuff. I won't put you on the spot. I won't ask you any questions. I hate that when you're in class and, who's a biologist? And they ask me the one question I can't answer. That's so embarrassing. So I keep my mouth shut. Okay? Now, let me show you. We're going to be here next, right? Now, I see what's happening. Carbon dioxide going off. I see another thing. So, what do you reckon will happen to that 5 carbon right there, down here? It's going to become 4. I don't really care about naming it. I just care about it. It's a 4 carbon compound. So let me click it and move the arrow to here. And there's the arrow down there. And this is this lower portion. And I'm right here working. So step 5 is oxidation. Remember the redox, the oil rig I told you about? Oil is, O is gain. R is no, rig, oil rig. Oxidation is, the G stands for what? Gain of electrons. And the rig part, reduction
means reduced.
I'm sorry, I got it backwards.
Yeah, reduction is gain.
Yeah, oil is loss. Reduction is gain. So we're going to see, he's losing. He's losing. That is oil. You're losing, right? So that's oxidation. So oxidation happens to this substance, which is a 5 carbon compound. Oxidized to make this 4 carbon Acetyl CoA. That's a Acetyl CoA, and you give off another carbon. That's why 5 became 4. What you really want to do with H's. That's what you want the H's. I got to remove the C so I can bust up the bond and let the H's free up. But I got to grab the H's. They're going to go out with the carbon dioxide and be breathed out too. So I got to grab them, and there's your grabber. It's called an acceptor. Okay. Now right here, now I got 5 carbons have become 4 carbons, and I don't really care about the name. I don't know myself. I know how the carbons go. Okay? I reckon names really mean a hill of beans just if you follow the carbon structure. Now now it's going to move over here. I'll point right there. And there it is, pointing right there. Now, remember we just made this one, right? You just made this one. It's a 4-carbon compound. Now, nothing's going to happen. Now, I did change it here. It looks like I did change it there. I just didn't change on my main one. But this phosphorus comes in. ADP comes up, joins the phosphorus, and you have produced ATP, which is energy. Energy. That's your battery. And you won't see it happen again. It'll happen one time. So Krebs Cycle only gives you one ATP. Only gives you one ATP. That's all it gives you. But I got it happening twice. I got 2 pyruvates, right? So I'm getting 2 in all, really. I'm just showing you one of the cycle, but it's happening side by side. Okay. Now, I didn't give a carbon dioxide off, did I? So the carbon 4 remains carbon 4, right? But because I have this coenzyme A was what's needed to make the phosphorus work. So when the phosphorus does the job, this thing drops off, and you have just succinate. Just still 4 carbons. Now my air is going to move here next. There it is. Now, again, I'm going to take succinate and make fumarate, which I don't care about the name fumarate. I just know it's still going to be a 4 carbon. I'm going to use FAD this time as my grabber. I'm going to give off 2 hydrogens and their accompanying electrons, and I'm going to grab them. And that's the compound, and you grabbed it. Nothing happened to carbons, but this compound has 2 H's more than that does because I gave away, right? So he is 2 H's less. So now we're going to move to here. Anything happen right there, you reckon? A reshuffling of cards. I'm right here. I'm just reshuffling cards. Just reshuffling cards. And fumarate is going to give him 2 H's. And then he's going to give up those 2 H's. It's going to transfer laterally. I do not know why it doesn't happen right there. You know, like all these did. But if I want to design this, I'm going to say, let's do it here and leave that alone. But that's not what happened, we discovered. Krebs found out that fumarate transferred 2 H's to malate, and then malate's going to give them to NAD. Don't know why, just how it happens. Okay? My area now is going to move right there. Now remember the 2 H's that we received? And now we'll give them off. Still 4 carbons. When I do that, I'm right back where I started. This can now go again. It is a cycle. And I have produced, there's 2 H's and 2 H's and their accompanying electrons. 2 more and their accompanying electrons. That's not. 2 more and their accompanying electrons. 2 more in theirs and 2 more in theirs. So I'm giving off a lot of electrons because when I give off the hydrogen, the electron tags along with it. And I want the electron more I want the hydrogen. And I gave off 1, 2, 3. Now right here it didn't require the loss of carbon to free those up. If you look back usually if you look back, and carbon comes off and shakes it up. See right there? But over here, there's no carbon coming off. There's no shake up. This has become so unstable. H is just glad to get off the ride. So I got 1, 2, 3 carbons coming off one of those pyruvates. But it happens twice, right? So I actually got 6 coming off. Now there's all my carbon dioxide. That's every bit that you breathe out. You breathe out 6 molecules, and it's all right there. When you breathe out, you're going to breathe out 6 molecules of carbon dioxide and 6 molecules of water. The water's going to come from the electron transport chain. When the electrons go through and hit the floor, something's got to grab them and carry them off. Oxygen does. And every 2 hydrogens that hit the floor and are finished, you got to get rid of them. And the H, those 2 H's join 1 O, that gives you H2O. And you also breathe out water too. That pyruvate is totally gone. It's totally gone. There's no trash left over. It's all gone. And that's why when you're breathing like this, there's no, you don't get tired. You know, if you're running, you can't consume enough oxygen. You have to stop and let this catch up. All right, now. For that, I finished that.
You finished 53.
I'm on 53? Okay. So, at this point, when I get here, I'm ready to start again. And that would be your 9th step. I don't know Krebs' first name. I saw a picture of him. He's a bald-headed man. But he was the one who actually laid out and described some of these inner workings. And because he did so much to explain the citric acid cycle, that other scientists have named it the Krebs cycle. And if you look up Krebs cycle and you look up, you're going to probably find both of them. But I did find a few more pictures under the citric acid cycle because there was no that longer. We had a longer history, so. And I found a lot of pictures on both of them. Okay, now, this is going to wrap it up a little bit. Glycolysis, remember? It made 2 pyruvates because it split glucose in half. Okay? So each pyruvate goes to the Krebs cycle. I got 2 of them, right? So the Krebs cycle cranks around twice. That makes sense so far. Okay? So everything happens in one of them, actually really happens twice per glucose. So one of them gives off 3 carbon dioxides, and it happens twice. Well, that's your 6 carbon dioxides, right? Everything is times 2 as far as the glucose is concerned. So for 1 molecule of glucose, I'm going to get 6 of these, 2 of those, 2 ATPs in all. And this means 3 NADs, 3 for 1 cycle, 3 of these, 1 of those, and 1 of those for 1 cycle. Now 2 cycles would be 2 times that. 2 times 3 is 6 NADs. 2 times 1 is 2 FADs. And 2 times 1 is 2 ATPs. So 1 glucose can produce still. It ain't too impressive, is it? Not thinking about 34 coming from somewhere else. All right, now.
Waste of time.
Yes, you got a question?
I said it's kind of a waste of time.
Yeah, but it's a waste of time except for the fact that you're getting a lot of what? What's the whole thing trying to give you? Why do you do the Krebs cycle?
Yeah, for electrons.
It's the electrons, and those are going to pay big benefits.
Yeah.
But you're right, as far as making ATP, it's a waste of time. It's almost like nature says, well, I gotta get something out of it.
Yeah.
So I'll get 2 ATPs, but you gotta remember now, the electron transport chain is gonna use these H's and those H's, produce 34 ATPs. Now that's a lion's share right there, baby. So, like I said, aerobic respiration uses oxygen. And this does 36. Where'd the other 2 go to? I said it's 38 and all. Where's the other 2 at? How does it all start? Glycolysis. That's your first 2. And then you go into mitochondria and get your other 36. Of those 36, how many come out of the Krebs cycle total? Just 2. So how many come from the electron transport chain? 34. How many times that chain run? Twice. Because each chain is going to give you half of 34. Everything is in half, remember? So when you get done, if I count the 2 from glycolysis and the 2 from the Krebs cycle, and these 34, the 2 plus 2 is what? 4. And those 4 plus 34 is, that's the max you're going to get from glucose. 2 from glycolysis, 2 from Krebs, and the other 34, the electron transport chain, which I'm about to tell you about. The next slides are about that transport chain. It's not really as complicated as the Krebs cycle was. Now remember now, I had 3 NADs from each Krebs cycle. I got 6 in all. I only had 1 FAD from one cycle, so I got 2 in all. But those 8 compounds, those 8 hydrogen carrying compounds, will then enter the electron transport chain. Okay? Now, the 2 Krebs cycles, they have all these electrons right here. And now, actually, we're done with the Krebs cycle. But these things represent energy I can use to build my other 34 ATPs. So, these 2 things right here, those 2 carriers, represent energy to do work. Now, the electrons are tacked onto those hydrogens, which are protons. You know how big that electron is? Let's say this thing is 1 nickel. Let's say it's 1 nickel. You take that nickel and you divide it into 1,835 pieces. Electrons are one of those pieces. You can't grab it, it's too small. I can grab that big old proton, knowing full well a plus charge and a negative charge, they will. So when I grab the proton, what's going to come with it? That teeny tiny electron, which is what I want. I don't want the H. I want the electron that he's married to. I don't want the H though. And when you breathe out water, all the H's, you're going to get rid of it. Because you didn't use the electrons in the electron transport chain. Now this chain happens on the intermembrane. What's that membrane called again? You labeled it.
It's
called the crystal. Now, the Krebs cycle happens in the water inside the mitochondrion. This transport chain happens inside that little skinny membrane. So the transport chain is linear. It's not circular, it's linear. And it does not recycle. You must supply new hydrogens to keep that circle going. If I strangle you and you die from no oxygen, for a little while, you can work with glycolysis, right? But when that happens and now you have nowhere to go and you can't get oxygen, respiration stops and you die. Now, the electron transport chain uses a ton of enzymes, which I don't know the enzymes. I don't make you know them either, but I know enzymes drive the electron transport chain. They all have names. Oxidized. If you oxidize, you gain or lose. Huh?
Okay.
Oil.
Yeah.
Is it? Yeah. Oil. Oxidation is loss. So you're going to lose the H's. See, this became that because he lost the H. That's called oxidation. When you gain the H, that's called reduction. The Krebs cycle did reduction. The electron transport chain is going to rip them back off and do oxidation. Now, I have no clue how to say that word. Ubiquinone, I guess. Q is what it's called. And there's another, that cytochrome C molecule. These are electron carriers. Here's my point. I got to strip this H off and this H off then remove the electrons and grab them. These are my electron grabbers. They are found only one place, in the crystal. Now the electrons, as they are passed from molecule to molecule in that membrane, they give off energy, which can be used to make ATP. Now, I've got a picture to show you, as you probably know. Okay. Now, the electrons are carried to the ETS, and they're going to provide the energy. It's going to pump hydrogen across the membrane. They're going to provide the energy to make it happen. The hydrogen is going to be pumped into that intermembrane space. In the outer area, there's a lot of hydrogen protons that could do work if you can make them move. The hydrogen protons, they move through an enzyme called ATPsynthase. What's synthesis mean?
Build.
To build. Guess what ATPsynthase is building? ATP. Okay? Now, from the outer compartment, they pass through like a turnstile where you go through and it cranks around and counts people. But it's going through that thing. Now, phosphorylation is where you're going to have that phosphorus added to ADP and make this. And the picture's going to clear it all up for you. And when we get done, I want to have 34 per. Okay? Now, when those electrons have finished with their energy, they combine with oxygen to make water. If oxygen's cut off, like a train to death, there's no place for the electrons and the hydrogens to go. They back up and they stop the show. Your life. And if you stop breathing, then there won't be a concentration gradient. The protons won't move, you won't make ATP, and you will die. And you die pretty fast too. It don't take hours, I mean, you die within a few minutes. Okay? Now, the electron transport chain produces 17. What's 17 plus 17?
34.
Ain't that 34? Because remember now, the Krebs cycle is only half the glucose, right? And each Krebs cycle has its own electron transport chain. Alright, so 17 from one Krebs cycle and that chain, 17 from the other one and that chain, there's your 34. Okay. Now when you add the two that you got from Krebs to these two, you're going to find at the end of mitochondrion, you get 36 of these bad boys. 36 of them. 38 counting the two glucose that glycolysis gave you. Okay? That's 18 times more than you get through fermentation. That's why a large critter, a large organism could not survive on glycolysis. Just does not enough energy produced. But a single cell germ can. A yeast is a single cell. Bacteria, single cell. They are small enough that 2 ATPs can keep them going. But big as we are, ain't no way. We've got to have aerobic respiration to give us all we need to keep us going. All right, let's look at the review of it. We're going to track one glucose, just one glucose, and you consume thousands of glucose. All right. Glycolysis, you know, where it ripped it in half, that gives you 2 ATPs. Okay. Now, that pruvate that you produce goes into anaerobic. Krebs gives you 2 more. 1 per Krebs, right? So far, that's 4. Then these Krebs H's go to the electron transport chain, and they're going to give you 34 more. So inside the mitochondrion, you're going to have 36. But if you count these two, you're going to see that you can totally get 38 ATPs from one glucose if you count glycolysis too. And you do. And you do. So when it's all over and done with, the glycolysis has been changed into carbon dioxide that you breathed out. I mean the glucose that you breathed out, into water that you breathed out, and a grand total of 38 ATPs. 2 from glycolysis, 2 from Krebs, and 34 from the electron transport chain. Okay? Now, let me show you a picture. I left the grids on, thinking it might help me explain this to you. But this is that membrane, this is the crystate. This is the crystate. All right. This right here is outside, and that's inside. And this is let me show you what I'm talking about. That's why I need to make a point. Let me find that picture of the mitochondria again. There we go. Okay, see the space right there? You see the space that's outside there? See, this is a membrane, and that's the membrane. There's space inside here too. See, that space in there. There's a lot more space inside, right? But there's space outside. The H's accumulate outside here. And then they go through here. As they pass through here, they produce those 34 ATPs. So I have the intermembrane, but what's intermediate between, right? Okay, this is the outer membrane. This is the outer membrane. It's outlined in red. And then this is the inner membrane outlined in yellow. This is the matrix. And this is the inner membrane area. There's room inside here for the hydrogens to accumulate. And by diffusion, things move across here. If there's 10,000 out here and one inside there, it's going to diffuse in, right? So you don't need any energy to make that. It's going to happen by itself. Let me get back where I was now. Okay. There's that space between the two membranes. This is blue on the other picture. This is blue area, other picture. Now, remember I told you that Krebs happens inside here? And it does. But look what happens. The H's are pumped out. They're released in here, pumped out. Now when the H leaves, this is the path taken by the electrons. That's the electron transport chain. And the electron, all they're doing is providing power to kick out the H's. This H won't move unless this electron provides the power to make it move. See, here's NAD. Okay, the H right there goes out. This H here stays inside. When you give off the H, you release energy. That energy, like swinging a paddle, is what drove this hydrogen outside here. It happens over and over again. I want the H's outside. I don't want them inside. I want them out there in that little skinny area called intermembrane space, because they're going to fall back through this sucker here, and that's going to make your ATPs. As the H's fall back through this is the ATPsynthase. As they fall back through, they themselves then produce the energy that's going to make this happen and give off this. If H's are not out here, can they go through that? If there are no H's out here, can this thing make ATP? No, because the H's must pass down through this way, and in doing that, they produce energy that can make ATP. All the electrons are doing is providing tons of energy to send the H's outside. It's the H's passing through this enzyme right here, because that's ATPsynthase, that's going to make the ATP. Now, what electrons for? They're going to drive the process of sending H's outside here so they can fall back through there and make the ATPsynthase. Without the electron, would this H go out here? No, it will not. Without the electron, would that H go out there? No, it will not. I need the electrons to give me the kick to send the H's outside here in this space. And that skinny, see, on the other picture, this is all blue, right? This is inside the mitochondria. So let me tell you again now. I don't care about the names of this, what it's called these things, but I just care one thing. The electron comes off here. The electron keeps traveling, gives more energy, keeps traveling. When it's over and done with, it's going to join these two to make water. So the electrons produce the energy to drive out the H's. They got to be outside so they can go back through this turntable. Now, there's so many outside here by diffusion, they're going to try to get back in. They're going to find the open door. They're going to pass through this thing right there. As they pass through it, it's going to spin like a turbine. It's going to make electricity. It's going to make power. And that power is used to take this phosphorus, add it to ADP, it's called phosphorylation. And then when these two join together, we have produced ATP, which is what keeps us alive. That's one picture. Here's another picture showing the same thing. I like to show different ways of doing this. In this picture, now, if you look carefully now, this looks a lot more complex. They're sort of the same thing. And these are different people talking. And this, again, there's your NAD giving off the hydrogen. The electron is going to take the hydrogen and make it leave the. So this right here is inside there. There's your matrix. So the NADH and the FADH also are going to release their H's. The electrons produce the power to kick the H's out here. So they can then, later on, re-enter. Just come right back around, go back in. This is a one-way street. It can't work both ways. They got to go this way to make that thing spin and make energy. So I have these complexes. All their job is to provide an avenue to send all these H's outside here in an intermembrane area. And all these H's start to accumulate, and they want to go back. They want to go back. Only way in is through there. That's the only way in. It's the open channel. And by George, as they pass through there, they spin this wheel kind of thing, and the phosphorylation occurs. This is ATPsynthase. Other picture call it coupling factor, because you're coupling these two. So you're coupling those. You hook them together like two train cars together. That's called coupling factor there. It really is ATPsynthase.
Are they leaving something behind when they go out too? That food that they're carrying?
What now?
When it says carrying electrons from food, are they carrying something else alongside of it when it goes out? No?
It's just pure electron. That's pure proton. The electron that he was married to is used right here to kick the husband out. The wife stays in and she boots him out. The wife be the electron. Here they are, they're married. At this point, the wife provides the boot to kick him out. The electron goes up, happens again. Another wife kicks them out. Another wife kicks them out. Now these are all the husbands. The wives are inside. Gonna come back in. Going to rejoin the wife. And there's your married couple there. They just split for a moment. And the wife is the one that's, like in most, she's the one that actually cracks the whip. Because the wife ain't happy, ain't nobody happy. And so that's why I use the wife and husband. I mean, he's going to do the work. He's going to do the work. She's going to make him do it. Another good simulation. When spring cleaning comes, I'm going to go fishing. Well, what do I do to keep her happy? I do the house cleanup. And like I said, this is a good similarity between a husband and wife. Here they're together. They separate. The wife kicks him out. Goes on. Another wife kicks the husband out. And now they fall back down, losing energy. Another wife, the other husband kicked out. The wives are in here still. They stay in here. Husbands are going to go back in. They're going to return and join the wife again and make the water right. But when the husband comes back, he's going to make something happen. He's going to make ATP. And that is the electron transport chain. What do you think is more complex, Krebs or this?
Krebs.
I think Krebs is more. Because without Krebs, will this happen?
No.
You got to have husband and wife, don't you? Without Krebs, this ain't going anywhere. But does oxygen get involved with Krebs or get involved with this? This. Because the oxygens, that's the way to finally get rid of those pesky husband and wives. The Krebs cycle produces a husband and wives and then sends those couples to the electron transport chain where they are split. The electron, I call the wife, will kick the husband out. That's the H. You have many husband and wives going over there, right? Yeah, all the husbands end up temporarily outside here. They want to get back to their wife so bad. They're trying to get back in, but that door's locked. There's the wall. Hey, there's a door right here. I think I'll pass through there. Well, they can pass through there, but as they pass, they spin this synthase. And in doing that, they take energy and put it right there. And this phosphorus then bonds to this, and ATP is formed. Now he is now free to go back and find his wife and make the water and get breathed out. That's beautiful. I mean, the more I understand it, I'm going, man, that is so cool. There's nothing wasted. No wasted steps at all. And I produce, good Lord, tons of ATP. Now, that's just one glucose. You consume thousands of a time. And the question, do you even need all the glucose that you really eat? That's what we call getting fat. And you eat more than you need, and we have more enough. So you would think a person would never be tired and run down, would you? But then again, they do. There are a lot of folks who have thyroid problems, and that makes them tired and run down because the thyroid produces two hormones that makes this happen. If they don't produce the hormones, this won't happen sufficiently. You don't get the energy, and I'm so tired, I don't do nothing, I just give out. It's all because of the thyroid problem. It can be hormones. And many drugs are hormones. That you take the drug and you feel better, you feel like going fishing today, whatever. Man, when I get up on Saturday, honey, I don't feel good. I think I won't go fishing today. She knows I'm serious. I go fishing down the path. I mean, take my leg off and go one-legged. But I like fishing. I like hunting almost as much, but fishing is more productive. Well, catch me a fish. I might not always get me a deer, but I'll catch me a fish. And the weather is so cold like this, I'm in fish withdrawal. And I want the spring comes, I can go fishing again. Okay? Well, that's the end of that slideshow. And that walked you through glycolysis, fermentation, both lactic acid fermentation and also alcohol fermentation. It walked you through what happened in the mitochondria if you get oxygen available. It told you about the Krebs cycle, how that produces all those hydrogens and electrons. It tells you how the electrons are then sent to the electron transport chain, which happens one place in the crystal. How they then use those hydrogen electrons to drive the ATP synthase with the hydrogen. It's all for one reason. make ATP. It's all it's for. Now, if you're running real fast and you don't have enough oxygen to use up the glucose, then lactic acid is going to accumulate, and you're going to give out, and you're going to just, I can't run another step, I'm so tired. And you keep breathing, because lactic acid is going to be turned back to glucose, right? And since you have oxygen now, that's going to go to the mitochondria, and you're going to use that totally up, and before long, you breathe in like you already are normal. And a person who runs a lot, their body gets accustomed to the added demands of oxygen, and they can meet them. A person who is a couch potato, well, I'm handicapped. I used to run all the time. Now I don't run at all. I know that my wreck happened in 2003. My lungs probably right now are nowhere near the condition they were before. I ran all the time. I like running. Turkey hunting, I run to the woods, not to catch a turkey, but to get point A to point B real fast. And I knew that my running gave me the ability to run longer before I hit the wall, so to speak. And you find a lot of people who play tennis, right now they're not playing tennis, and then spring comes, they overdo it. You know your muscles get sore, don't you? Well, they get sore because you produce lactic acid. And the lactic acid accumulates there in great numbers, and that causes swelling, and that's soreness. If you keep exercising, what will the soreness do? It will go away. Now what's better? 1,000 reps of 1 pound or 1 rep of 1,000 pounds? 1,000 reps. It's better. It's not so much how much, it's how much you make the muscles work. So my wife has the 1 pound weights. She uses it all the time. She has fibromyalgia. And when she don't do exercise, her joints hurt. And then I said, now come on now. And I'll get on the floor and I do my exercise and I'll wrestle sometimes. Well, one leg now, she can beat me. We wrestle. I don't throw her down, but I'll grab her by the foot and do it. Sometimes I chase her, sometimes she let me catch her. Did I know what I do? Anyway, when I come back and see you next time,